Okay, so today's lab starts on page 45 in your packet, looking at the digestive system. So we're going to look at some chemical, mechanical processes of a digestion. We're going to look at the different tissues that we see throughout the digestive tract, starting at the esophagus, ending with the large intestine, and then looking at the blood supply that goes to the digestive tract, and looking at some of the accessory organs like the pancreas, the liver, the salivary glands that aid in digestion, and then looking at specific parts of the hepatic portal circulation, which involves the liver. The liver plays a major digestive function in the body, and we'll talk a lot about that at the end of lab. So let's start out with by just differentiating mechanical from chemical digestion. And I do have a PowerPoint for today's lab. It is, okay, so this is the PowerPoint. So we're going to start with looking at the tissues of the esophagus, and then we're going to see how that changes a little bit in the stomach, and then how the first part of the small intestine has some changes structurally as well. It's called the duodenum, is the first part of the small intestine. So we're going to start up here in the esophagus as food enters into the mouth, and we start to swallow it as it enters the esophagus. No, that's not the throat. The esophagus is below the pharynx. The pharynx is the throat, but the esophagus is strictly for food. So we have the trachea, that's for air, and the esophagus that lies behind it, which is destined for food. So because we're not always eating, but we're always breathing, right? The trachea is always open. It's always held open by cartilage. So when you look at a trachea slide, you're always going to see a wide open lumen. But when you look at an esophagus slide, it's going to be collapsed in the center, like we can see here, the white space, it's collapsed because there's no food in it. We don't need a expanded esophagus when we're not eating, so it lies behind the trachea and it's collapsed when there's no food passing through it. So because the food we eat is sometimes rough and has, you know, abrasive materials in it, like when you're eating roughage, an apple, some Doritos, tacos, that could really do some major damage if we had a thin layer of cells lining the esophagus. So what we find here is that layer of cells that are protective in nature, and that's stratified squamous epithelium. Stratified means we have many layers, and squamous means many layers of flattened cells. Squamous, think squashed, flattened cells. So many layers of flattened cells line the esophagus. And then outside of that, we have a smooth muscle layer. And the smooth muscle layer is what helps move the food along. So you see this muscularis externa. This is where we see those wave-like contractions, peristalsis that moves the food toward the stomach. So once the voluntary part of swallowing is over, then we have the involuntary process of just moving that food toward the stomach. So if you hang upside down eating a peanut butter sandwich, that swallow, once you swallow it down, it'll make its way to your stomach even though you're hanging upside down because of the peristaltic waves moving it in that direction. So if we look at the stomach mucosa, the inside wall of the stomach is made up of these little holes. See those holes? Those are called gastric pits. It just provides greater surface area for digestion when materials move down these pits. But a majority of the top layer of these pits is mucus. Because what do we know that the stomach secretes? That gives us trouble sometimes if we eat too much junk. Acid, right? We get acid reflux because the special cells of the stomach that secrete hydrochloric acid. So a major secretion in the upper portion of these cells over the surface of the stomach is mucus to protect the stomach from digesting itself. But as we age, mucus production goes down, but hydrochloric acid production doesn't. So as a result, we're more at risk for ulcers as we age. So that's why people, as they age and get older, they don't like to eat spicy foods and things that are a little more harsh on the digestive process. Or if you've eaten a lot of stuff and the digestive processes kind of stop, you might find that you feel like your stomach is still full like 2 hours after you've eaten a meal. Did you ever have that where you go to bed and it's like, ooh, this is not breaking down, right? That's because acid levels are high and some of the enzymes in your stomach and small intestine don't work when acid levels are high. So digestion does stop. And sometimes you roll over and you feel that liquid kind of roll in your stomach. That's the worst. Well, taking an antacid What's that going to do to the pH? Antacid. It's going to bring the pH up. It's going to help those digestive enzymes work, and then digestion kicks in, and half an hour later you feel better, right? And then we go for more food, like at Thanksgiving time, right? But the stomach mucosa protects itself with this layer of mucus, and a lot of the digestion and secretion is down in these gastric pits. So if you look on the surface, all these little holes here are called the gastric pits. And if we look a little closer, here's a gastric pit, and then we have these simple columnar epithelial cells lining the pit. And there's different cells, so just because they all look the same doesn't mean that they have the same secretions, because we're going to look in detail, particularly in lecture, on the different cells that have different secretions down this pit. The ones up at the surface, a little higher up, their job is to secrete the mucus, again, to protect the stomach lining so it doesn't get damaged. So simple columnar epithelium, simple meaning we have one layer of column-shaped cells. So this is what we find throughout the digestive tract. Once we get out of the esophagus, we see simple columnar epithelium because we don't have abrasion going on anymore. We want absorption and secretion. There's a lot of activity, a lot of enzymatic activity. Anytime we talk about enzymes digesting, that's an example of chemical digestion, because you have a chemical digesting a chemical, right? The food we eat, carbohydrates, fats, lipids, nucleic acids, those are all chemicals, right? Made up of atoms bonded together. And enzymes are what chemical class? Proteins. So these proteins are acting on these chemicals we eat and changing and breaking apart bonds and releasing energy and forming new ones and so on and so on. So that's chemical digestion. So objective number one asks you to differentiate mechanical from chemical digestion. Well, chemical digestion, yeah, I want you to think enzymes. Enzymes are acting on the food. Mechanical digestion is the crunching and the moving of food back and forth to break it up into little pieces. So just think of mechanical, like physical movement of food in the digestive tract. So mechanical digestion starts in the mouth with the help of the teeth when we chew the food. Wouldn't you agree that would help begin the digestive process, because it's breaking up the food, exposing it to your saliva, breaking it into smaller pieces? So when we chew our food, that's mechanical digestion, and that's called mastication, just chewing. And then as it enters and then rolling around with your tongue too helps with digestion, right? You roll it around for a while, chew it a little bit, roll it around with your tongue, chew it a little bit more, and then you send it down your esophagus. It hits the stomach, and the stomach pushes it back and forth. Its job, just with the muscular contractions of the stomach, it moves it toward the pylorus, then back up toward the fundus, back and forth, back and forth. That's mechanical digestion. That's the main function of the stomach, is to mechanically take this bolus of food that you mushed up in your mouth and swallow down and turn that into a liquid. And that liquid is called chyme. C-H-Y-M-E is how you spell it. But we'll talk more about that in lecture. So we have the simple columnar epithelium. We have the gastric pits. And then the gastric glands are essentially the cells at the bottom of this pit secrete hormones. Some of them do. And we call them gastric glands. So just think of gastric glands are at the bottom of the pit. So like I said, some of these cells, even though they're all simple columnar, they have different jobs. They secrete different things, and we'll talk about that in lecture. But for lab, all you have to know is that they're simple columnar cells, and when we get down to the bottom, we call them gastric glands. So if you see something here, an arrow at the surface, we call it a pit. If we're looking down deep into the pit, we call it a gland. So all of these would be glands. And up here at the surface would be a pit. And like I said, these are all mucus secreting cells. So looking back up at the surface, there's the pit, all these pits. And then remember, there's also wrinkles in the muscular wall called rugae that allow the stomach to stretch. So when it's not full of food, it's wrinkled down in a much smaller size. And then it stretches as often as we stretch it with our food. So the more we eat, the more we stretch it. And if we eat large meals, like some people don't eat it all during the day, they have a busy job. Maybe you're working in ER like Hunter, and you're having a busy day. There's some big thing that happened, and you're busy, and then you get home and you're starving and you eat a large pizza by yourself. That happens. People have done it, right? You stretch your stomach really big when you do that. And then if you do that on a regular basis, that's what leads to obesity, because that stretched stomach, when it starts to shrink down, says hungry, 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 when actually it's, you know, been stretched beyond its normal resting length. But our stomach, we're going to find out in lecture, has an ability, smooth muscle has an ability to have a new resting length if we keep stretching it. And that's how people get to be 1,000 pounds, because they eat very large meals over and over and over again, and their stomach is stretched that it can accommodate that food on a regular basis. So what do we know about healthy eating and healthy living? Small meals at regular intervals throughout the day, right? Keep your stomach at a normal size, keep the fuel in the furnace burning. And that's just a simple way to eat in a healthy way. Okay, so moving on then, here's just another view of, here's the lumen of the stomach. This is a cross section of the stomach, and here's the gastric pit right at the surface, and then down here is the gland, and you can see these simple columnar cells. And then we have muscle, smooth muscle found outside of that columnar epithelium. So out here, well, it doesn't quite show it. Let's go a little further. We're in the small intestine. But we have smooth muscle beyond the pits, and that's 2 layers of muscle in the stomach that helps move that chyme back and forth, breaking up that food, like I said, into a liquid. And we see a little bit of protein digestion occurring here. So that's when we eat some protein, that does begin to digest in the stomach. And then if we get to the small intestine, we see the same thing. We see these simple columnar epithelial cells. And inside of these columnar cells, we'll find goblet cells. So goblet cells secrete mucus as well. We see mucus secretion occurring throughout the tract, intestinal tract. So these are goblet cells secreting mucus. But the inside lining is kind of like the opposite of the stomach. So the stomach has these indentations that are these gastric pits, right? The small intestine is the opposite. It has these little extensions of the inner wall that are finger-like extensions we call villi. So this is a villus. This lines the small intestine. So you see these finger-like microvilli lining the small intestine, and these are all simple columnar epithelial cells. So they absorb the contents from the digestive tract, and then from there it goes into the blood and also goes into a special vessel for fats. So fats are processed by the small intestine and sent into these little structures, this green structure called a lacteal. So this is necessary for breaking down fats in the digestive tract, this green structure called a lacteal. And then the rest of the material enters into the capillaries. So again, this is a villus. This whole thing is a villus, and this is a simple columnar epithelial cell. Here's another.
So the lacteal breaks down the fat?
Mm-hmm. Yep, it's inside the villus. So here we can see all the villi of the small intestine. You can see the individual simple columnar epithelial cells that line the villus. And then deeper down below the villi, we see the smooth muscle of the small intestine, again, which is going to help, you know, bring that food through the digestive tract via peristalsis. So this is the smooth muscle layer, and these are the villi. And what are the white structures? Yeah, goblet cells that secrete mucus. So the small intestine is the main structure for absorption and digestion in the digestive tract. So the small intestine is hugely important for digestion and absorption. So these multiple cells lining the villi really increases surface area. And if you remember from general A&P, if I look at a simple columnar cell of the small intestine, if I just pull one of these little cells out, I'll see that it also has microvilli, where the plasma membrane has been pinched up into these tiny little projections that further increase surface area. So it's just really well designed to increase surface area for absorbing nutrients from the small intestine. And then if I go a little deeper, I find these Peyer's patches. Peyer's patches are part of the immune system. They're part of what we call mucosa-associated lymphoid tissue. So it's part of the immune system, and what it is let's say I ate some infected meat and I absorbed some of that into my blood, and that made contact with the inner layers of the small intestine. We do have some immune function here to help fight infection from things we eat in the digestive tract. So not only in the blood will we have, antibodies and immune cells to help, attack those foreign invaders like T cells. We also have these little clusters of immune tissue deep in the small intestine called Peyer's patches. So when you look in the slides today, pay attention to those. They're beyond the mucosal layer down here in the submucosa. Okay, look for those clusters of kind of dark tissue. Those are the Peyer's patches. You do need to know those. So then when you get down to the large intestine, we have, again, this columnar epithelium, smooth muscle beyond that. So here we can see the smooth muscle. And then we have what we call these crypts. And these crypts are just extra spaces. So these crypts are deeper indentations of the large intestine, because the large intestine's major secretion is mucus. So that's the job of the large intestine, is secrete mucus, which helps move things along, keep feces moving throughout the bowel, and to reabsorb water. Because we know if stool doesn't spend enough time in the large intestine to absorb water out, and someone gets the diarrhea, they're at risk for dehydration, right? Because that stool didn't stand there long enough to reabsorb that water and put it back into the blood. So it's very important anytime someone has a bout of diarrhea that they're rehydrating, because that fluid that they're losing in the toilet was meant for the blood. And we also know the opposite is true. The longer stool sits in the bowel, the more moisture that is absorbed out, and the stool becomes very hard and dry. And the muscular contractions of the large intestine keep moving the stool and moving the stool until it becomes a hard ball, like rabbit pellets, right? When you look at your patients that have been constipated for a long time and they're passing small, hard, round stools, they're pretty constipated. So it's important that we provide enough fluid in the diet, that we're encouraging water, we're giving them, foods that promote bulk, give bulk to the intestine. And if necessary, give them a laxative just to get things moving. Prune juice is a great stimulant for the large intestine. And one thing that works really well in younger people, I sometimes have to remind myself to be careful because I did this to somebody. I was so used to having 80-year-olds with slow bowels, I would give them the prune juice with butter, and I think it was just prune juice and butter, actually, and then you warm it up. And something about the warmth and the fat with the prune juice really kicks in the bowels for some people. And it wasn't working for my 80-year-old, so I had to put a little Miralax in theirs to get theirs going. Well, then I was just kind of on a roll this day, and I had a guy that was 60 who hadn't had a bowel movement in a week, and he didn't want to talk about it, but he hadn't had a bowel movement, and he was miserable. So I gave him that, and it kicked him in 30 minutes, and he had to cancel his appointment for therapy because he had the diarrhea for a couple hours. But it worked, and I said, hey, you can always reschedule therapy, but don't you feel better? All right, so moving on. And you can't underestimate the importance of the bowels. If you've ever been constipated, you know how miserable that feels and how it really kind of takes away your energy. It takes away your ability to eat more food and keep your energy up because things aren't moving. So it's important that we address bowels in our patients. There was someone in one of my clinical experiences that I heard about that actually died because they were on a feeding tube and they were had a traumatic brain injury, so they were on a ventilator, and no one watched the bowel movements, and they kept giving him feeding tube, perforated his bowel, necrotic bowel, died. So we have to remember the bowels. It seems like really low-level nursing and intervention, but it can be deadly if we don't take care of it. And I've had stubborn patients that say, nope, I don't want any laxative. I don't want that. No, even though it's been 2 weeks since they had a bowel movement. And the doctors will be very direct and say, you're here for your heart, but you could, leave here because of a perforated bowel on, going to the morgue if you don't let us take care of this. So it's very serious. And as nurses and caretakers, like at the nursing homes, there's a bowel record, right? If they hit day 3 and they haven't gone, we're gonna do something about it because it's very serious. Gotta watch that. Always let that be something you look for when you go to start your day. Look and see when their last bowel movement was, because it's very important that we watch that. And people don't want to talk about it because it's embarrassing.
It's part of the accessory portion.
It is, exactly. All right, so then moving on, looking at the intestinal villus. We kind of talked about that already. Do I have a picture? I had that one up here. This is objective number 3 on a model or diagram. I'm going to actually go to the Blackboard site. I do have a model of the small intestine right here. So if we look at this diagram, here we see the blood capillaries are inside here. And then we have the green structure, that's the lacteal. And the blood capillaries, like I say, are inside this. And what is this whole structure, this whole finger-like structure called? A villus. So this yellow one, that's the outside. We're just looking at a cross section here. So these are all villus. And then the microvilli, again, is one individual small intestinal cell, one columnar cell that has its plasma membrane kind of pinched up into small little extensions called microvilli. Another name for the microvilli on the simple columnar cells of the small intestine is called the brush border. So each of these is a villus, but each individual cell also has little extensions. Again, we call microvilli. So at the bottom of your Blackboard page, you'll see this picture of the brush border. So this is one simple columnar epithelial cell lining the villus, and this is the plasma membrane of that simple columnar cell. So microvilli or brush border. One cell, plasma membrane pushed up into these little finger-like extensions. So just think of villi are the big ones on the model, and then microvilli are the individual columnar cells lining the villus that have those little finger-like extensions. Okay. All right, so back to our objectives. So looking at a microscope slide of the accessory organs now, so the salivary glands. So salivary glands are these cauliflower-like structures that we see embedded around the muscle and bone in the cheek. We find them in front of the ear. Remember, you had to know these for general A&P, the parotid salivary gland, the sublingual under the tongue, and the submandibular under the lower jaw. So if we do a cross-section of these, we find these ducts. Those are the open centers. The duct are the passageways, right? So they secrete the saliva. And then the glands are the cells that make the saliva. So these are where the secretions are made, and then those enter the duct. So when you look at the slides, the pictures, the open center is the duct. Again, the cells are the glands. So the name for the gland, though, is not salivary gland. You have to call it alveolar gland. So be sure you know alveolar gland are the little cells that line the open ducts. And if we look at the pancreas has a number of functions. So letter C on number 4, page 45, talks about exocrine and endocrine. Do you remember what endocrine means? Where does that secretion go? It goes to the endocrine, means to the blood. It goes to the blood. So endocrine, the endocrine function of the pancreas is whatever it secretes to the blood, which has system-wide effects. Exocrine means that it's going to some duct that's nearby, and it's just going to do its secretion nearby. So exocrine, endocrine function of the pancreas. So that what does the pancreas secrete to the blood that diabetics need help with? Insulin. And then what is the opposite of insulin? Glucagon. So I have insulin to lower my blood sugar, and I have glucagon to raise my blood sugar between meals, right? We don't all pass out if we wake up in the morning, run to class without breakfast because of the help of glucagon. It raises our blood sugar. So that's secreted to the blood. So those cells that secrete insulin to the blood are called islets of Langerhans, or beta cells. So if we look at, do we have a, no, we don't. Okay. So looking at the exocrine functions, we have the Accessory cells, which secrete digestive enzymes. So the digestive function of the pancreas is to secrete digestive enzymes. So these are more numerous. You'll notice that these are much more numerous in the pancreas, the Accessory cells, the Accessory. So all these cells, you can see the nuclei, these all release digestive enzymes to the pancreatic duct. So here's the pancreatic duct. Can you see this open center here? These are the cells that line the duct, but this duct picks up the digestive enzymes secreted by these Accessory cells. These clusters of cells over here, they're lighter in color and they're in clusters. Those are the pancreatic islet cells, and they're made up of beta, alpha and beta cells that secrete glucagon or insulin. So you don't have to identify the alpha and beta cells of the islet cells, but you do have to know alpha cells secrete glucagon, beta cells secrete insulin. Because if you look in objective number 6, it says give a function for those of the following. So letter C, the pancreatic islet alpha cells secrete glucagon. That's number 6C on page 45. And 6D, the pancreatic islet beta cells secrete insulin. So now we'll talk about the liver, because there's a lot going on here in the liver. I think it's kind of cool, actually, this diagram, and there's a model as well. So the liver is made up of these hexagonal units called lobules. So if we go to the blackboard, here we can see this model here. So this is, each one of these hexagonal units is called a lobule, so you need to know that. And a lobule is made up of layers of hepatocytes, which are liver cells. So layers of liver cells kind of piled on top of one another are called lobules. So it's the structural unit of the liver. So going back to PowerPoint here. So each of these individual cells here is a lobule. So you can see is a hepatocyte, not a lobule. Each individual cell is a hepatocyte. Hepato means liver, cyte means cell. So these are all liver cells. Again, arranged in these little structural hexagonal units called lobules. So within them are these, let's look at this one here. We have these grooves within the lobules that help distribute materials throughout the cells, which are called sinusoids. So here we can see the sinusoids here. So what happens essentially is we have a special vessel that comes from the outside and it works to the inside. So blood from the digestive tract. So we absorb this food and it goes into our blood from the digestive tract. It goes to this what's called the hepatic portal vein or circulation. And remember when we talked about the portal system? Where else did we see a portal system? What two organs had a portal system? Everybody's pointing to their head. You're right. The hypothalamus and the Pituitary gland. Yeah, the pituitary gland. There was a portal to the anterior pituitary, right? So there's that portal system, which means it's a special connection that it doesn't go to the general blood supply. It goes straight to another organ. So what happens is when we digest things from the digestive tract, it goes straight to the liver through the hepatic portal vein. So the vein carries blood away from the digestive tract but takes it to the liver. And then it travels through these branches of that vein called sinusoids to expose it to the hepatocytes. And the hepatocytes do its job of metabolizing and helping break down the contents of, metabolites that we might have in the blood. But in the digestive tract, its job is to couple of things. One is fight infection of things that may have breached into the blood that from something we ate. There's these special cells inside here, and they're called Kupffer cells. They're in the sinusoids that help fight infection as blood flows through them. We also have oxygenated blood coming through here to feed the hepatocytes, because these need oxygen and nutrients to survive themselves, right? So we have freshly oxygenated blood coming from the hepatic artery also flowing through here. And then opposite of that, the hepatocyte's job is to secrete bile. That's the liver's main function, is to make bile to break down fat. So bile is going in the opposite direction. It's going away from the inside of the liver and going out to these small little canals, which we call canaliculi. So you'll see there's bile canaliculus. Those are inside the, kind of buried among the hepatocytes. So if we go back to our Blackboard site and look at these models here, I can get a little closer and see if I can look inside here and make this bigger. No, maybe not. No. Okay. But anyway, here you can see the purple are the liver sinusoids, and the light green you see here, I'll put a bigger picture online so you can see this up close. But the little light green structures, those are the bile canaliculi, and that leads to a bile duct on the outside. Eventually, they'll lead to the bile duct, common bile duct, or the hepatic duct for now because it's still in the liver. And then once that blood is filtered and the bile is secreted, it works toward the center, because remember it comes from the outside. Let's go back to the PowerPoint. So this blood is coming from the digestive tract, needs to be processed by the liver, and then it flows, all these sinusoids flow toward the central canal. So the central canal or central vein is what your lab, we're on objective 4 on page 45, B4, leads to the central vein. This is going to eventually lead out to the inferior vena cava. So this is blood that's been processed by the liver and it's ready to go back to the general supply. So here, if we look from the outside view, so here's the digestive tract. There's all these blood vessels that are leaving the digestive tract, leading away from the digestive tract and going to the portal vein, leading to the portal vein, and then heading to the liver for processing. So if we look at the different vessels, this is objective number 7. We have the gastric vein. Whenever you hear the word gastric, gastric bypass, gastroesophageal reflux, gastra means stomach. So gastra, stomach. And this will help you when someone says they have a nasogastric tube. Where is the tip of the tube? Extend the stomach. What if they have a nasoenteric tube? The tip of the tube, entero means intestines. So entero means intestines. So if someone has a nasoenteral tube, they bypass the stomach and they kept going, and the tip of that tube is in the small intestine, and we do see those. So that's going to help you. And also, if I tell you what is the organ that secretes gastrin? The stomach. Easy, right? Gastra. So keep that in mind. That will help you all the way through some of the programs you're going to be in, knowing those prefixes, and it kind of gets you to the right organ and then helps you kind of sort your thoughts. So entero means intestines. So if someone says that they are going to give their patient parenteral nutrition, that means the nutrition is not in the intestines, it's next to it. It's in the blood. It means we're giving nutrition via an IV straight to the blood. So what are we giving them? Do we give them the chocolatey stuff in their blood? No way. We give them the basics, right? We're going to give them electrolytes and sugar. And, you know, it's pretty much it. We're not going to, and basic vitamins, but we're not going to give them the chocolatey looking liquid that we give in feeding tubes. Okay, so looking at the blood supply then. So the gastric vein comes from the stomach. So just look over the vessels of the stomach. That's the gastric veins. The splenic vein comes over from the spleen, which is behind the stomach. So the gastric veins are here. Then we have the splenic vein. Then the superior mesenteric is a larger vein because it drains most of the intestines, small intestine, large intestine. So this is the superior mesenteric vein. The inferior mesenteric vein is much thinner. It only supplies, you can see the descending and beyond of the colon. So the inferior mesenteric vein, much smaller than the superior mesenteric because it supplies most of the digestive tract. The hepatic portal vein, like I said, all of these veins lead to the hepatic portal vein, leading to the liver. So this large kind of common trunk of these veins is the hepatic portal vein. So you have, again, the gastric vein coming off out here, the splenic vein at the base, inferior mesenteric, superior mesenteric, and then the hepatic veins are the veins that leave the liver and head toward I don't think that's shown on here. Hepatic veins leave the liver and lead to the inferior vena cava. I'll give you a page reference for the hepatic veins. And this would actually be in the blood vessels chapter of the circulatory system. That is shown. There's a really nice diagram of all of these veins on page 749 of your textbook. 749, figure 1929. And unfortunately, the older versions of the book even changed up the figure numbers to force everybody to buy the most recent edition. Tricky, huh?
What was the figure number?
Figure 1929. I'll go find it. All right, so again, superior mesenteric, very large vessel, inferior mesenteric, much smaller vessel. Just kind of know how they branch. You can see the gastric veins come off first and then the splenic vein. So gastric veins, whichever, yeah, whichever pin is on. And then the splenic vein comes kind of off behind the stomach. That's why it's faded out here, because it's actually behind the stomach, because that's where the spleen is out here. And the spleen is really important. It's part of the immune system, has a very rich blood supply, so it has a pretty large vein you can see there, because it's a very vascular organ. When people get in traumatic accidents with, blunt force, like accidents where they smack into something or they get rear-ended, sometimes the spleen will burst and then they lose their spleen. I'm sure Hunter has seen that, being someone who works in an emergency. So that's your hepatic portal circulation. So going back to our objectives then, we gotta go back to our blackboard. So we talked about, I believe, all the structures. One thing that you need to know, though, is looking at the end products of digestion of the different nutrients that we take in. So in your textbook, there is a table, and I also put it on Blackboard in the lecture section though, but this is going to help you with this part of the lab as well. So I'm going to go over here to the lecture and know this flow chart really well, which means that if I bring in a starch or a sugar, the enzyme that's going to act on it is brush border enzymes in the small intestine, salivary amylase, pancreatic amylase, and it's going to break it down into what? Starch and disaccharides are going to be broke down into those monosaccharides, right? Fructose, glucose, galactose, those are the monosaccharides. That's the end product. Once all the digestive enzymes have done their job, this big long starch molecule is going to be these tiny little monosaccharide sugar units. If I bring a protein into the body, what's going to attack it? Pepsin, pancreatic, you don't have to know the names of the enzymes, it's not part of lab, but just know that what's going to break it down? Where does protein digestion occur? Stomach, small intestine is going to break it down into amino acids. If I bring in fats, what's going to break it down? The liver is going to secrete bile, right? The small intestine is going to have those lacteals present, right? But there's also digestive enzymes called lipase. It's going to break it down into fatty acids and glycerol. So the end product of digestion is fatty acids and glycerol. And what's helping in that process? Small intestine and the liver. And then nucleic acids are going to be broken down into the pieces of a nucleotide. Remember, we have the sugar, the deoxyribose or ribose sugar, and then we have the phosphates, and then we have the nitrogen bases. And again, the pancreas and the small intestine. So small intestine is throughout, right? So when in doubt, definitely include the small intestine as one of the organs involved for absorption and digestion. And then with fats, you want to add the liver in there, protein, add in the stomach, and starches add in the mouth with that salivary amylase. That's a component, that's a digestive enzyme we see in saliva that starts starch digestion. That's why you can give a baby with no teeth a saltine cracker and it'll just turn to a liquid in their mouth. But if you give them a tortilla chip, it's going to stay there because it's not going to break it down completely. So be sure that you reference this chart also. This is in your the lecture folder, like I said, because I'm going to reference this in lecture as well. Any questions? That concludes the digestive lab. So on the back side of your objectives then on page 46 at the top, there is a little extension that you do need to know the digestive enzymes and the substrate. So the substrate is the food particle that we're talking about. So if we look at the table in your lab packet, so I'm going to go up, this is copied backwards, but here's the table. So the substrate are the different types of foods we eat, right? Carbohydrates, proteins, fats. Those are the substrates. So you have to know the enzymes for carbohydrates. So the two enzymes you have to know are amylase and disaccaridases. And the enzymes for fat digestion are lipase. And then for proteins, we got quite a few. We've got pepsin, trypsin, chymotrypsin, carboxypeptidase, and peptidases. So again, the major site of enzyme activity, you have to know where they come from. So salivary amylase, we find that in the oral cavity coming from the salivary glands. Pepsin comes from the stomach. The duodenum is pancreatic amylase because the pancreatic duct leads to the duodenum. Trypsin, chymotrypsin, those come from the small intestine again, or it acts on the small intestine. Where does it come from? It comes from the pancreas. So if you look at where a lot of these three different groups of enzymes, they all come from the pancreatic acinar cells. Peptidase, disaccharidase comes from the small intestine, and where does it act? In the small intestine. So most of the action where these enzymes are secreted to is in the small intestine. So when in doubt, choose the small intestine. Except for stomach, we see a little bit of protein digestion in the stomach. And then the oral cavity for the amylase. So just pretty much knowing this table is going to help you through those couple of questions that cover this objective.